Hello everyone. If you are watching this video, it means that I had an emergency and I knew that I had to take a child to the doctor's office at 7:30 this morning, but I was really hoping that I would be done in time to show up for class, especially since I haven't seen you guys in a while. But the fact that you're getting this video means that I am on my way back, and I really didn't want you to just sit and wait for me. So what I'd like for you to do is pause for a few minutes and just turn in your assignments to Daniel if you haven't done that already. I have asked Mr. Meese, and you remember him from the ACE lab, to go ahead and fill in for me for about 10-15 minutes until I get there. So while y'all are waiting for me, go ahead and turn in your assignments to him. And after you've done that, I want to talk a little bit about the genomics and biotechnology chapter, because a little bit of that is going to be on your test also. And then hopefully by the time you finish watching this and get a little bit into your review, I should be back in class. So remember that your test is going to cover protein synthesis, gene regulation, gene expression, and then biotechnology and genomics. So it's not a whole lot on it, but I think you guys deserve a break. So the homework that you just did was over chapter 17, and I sent you a video to watch, basically talking about what you do with DNA. What you guys did in lab yesterday, and those of you who have lab today are going to do, is you are going to actually extract DNA from some cells, and you guys extracted it from plant cells, and I think you had fun doing that. But that's basically what scientists do, and they get DNA. But what do you do when you get DNA? Well, the next step is usually to sequence DNA. And so when you were doing your homework and watching the video, you looked at how do you sequence DNA. When you sequence DNA, one of the things I want you to remember is the difference between genomics and proteomics. So you remember that when you're looking at genomics, you're looking at the entire genetic makeup of an organism. So all the genes that make up an organism is known as genomics. When you're talking about proteomics, you're talking about all the proteins that make up an organism. And so the genes, of course, are going to be much, much more, because you have a lot of genes that sequence, or genes on sequences on DNA. So you have all these nucleotides, and those nucleotides are going to code for the different proteins, which of course are made up of amino acids and all of that. You guys know all that process. But I asked you to look at the difference between genomics and proteomics, but also know that you can divide genomics into different sections. So you should also know the difference between functional genomics, comparative genomics, and metagenomics. Functional genomics means I'm sequencing a DNA, and sequencing a DNA means how do I know which nucleotides are on which position of a chromosome? That's what I want you to be thinking in your mind when you think of sequencing of a DNA. So how do I know the DNA sequences, the nucleotide sequences on this person's chromosome? I know that you have 23 pairs of chromosome, and I know that the first pair are going to be your, so somebody help me out. Okay, you're looking at chromosomes and you're looking at a karyotype, and you remember, are those your metacentric or your submetacentric? Those are your metacentric chromosomes. So I know that my chromosome 1 are my metacentric chromosomes, but how do I know what nucleotides are on each of those chromosomes? What I do is sequence them, and when I sequence them, then I know what my nucleotides are. So why do I sequence them? Well, sometimes functional genomics means I'm sequencing this DNA because I want to know what the function of the specific gene is. What does the gene do? Okay, that's functional genomics, trying to determine the sequence of the function of each gene in this sequence. And then you remembered metagenomics. Metagenomics basically looks at the environment in which the gene is found. So I don't know the sequence of an unknown. I have a chromosome and I don't know what it does. Well, if I look at that organism and I look at all the other organisms that are found in this environment, if I know the sequence of DNA on the other organisms in that environment, then I can draw a conclusion about this organism. So if there's DNA that is found in my stomach cells and I don't know what that DNA does, by doing metagenomics and looking at the DNA of all the other organisms that are in my stomach, then I can say, okay, this is what this DNA does. Okay, that's metagenomics. You're looking at the environment in which one genome is found, or you're looking at the environment in which one organism is found, and you're comparing those, which leads you to the next one, comparative genomics. So comparative genomics now says if I look at an organism and I know the genome of one organism, I can compare that organism to other organisms and know that the relationship between them is determined by how similar their genomes are. Okay, so let me see if I can say that again. Metagenomics means look at the genome of an organism, but look at the genome of other organisms in its environment. Comparative genomics says look at the genome of an organism and then see the relationship between that organism and other organisms that have similar genomes. So you're comparing them, but you're comparing them to find a relationship. Metagenomics basically says you're not looking at the relationship between them, you're trying to look at it and compare it with another one that is found in its own environment. So environment, metagenomics, functional genomics, comparative genomics, relationship. That's the one that people look at evolutionary relationships, and they say, okay, if your genome is very close to the genome of another organism, then evolutionists would say that you guys are more closely related, or you guys descended from the same organism. Okay, that's basically that. Another thing that I wanted us to talk about as you get ready for your test is this whole idea of biotechnology. So when you're looking at biotechnology, what you're doing is you look at the genes of an organism, but you look at how can you use some techniques to study the DNA of an organism. So biotechnology is basically the science that deals with studying of DNA using some molecular techniques. So you probably have heard of genetic modified organisms. That's an aspect of biotechnology. What biotechnology does is look at a DNA, study the different parts of the DNA, and then be able to manipulate that DNA. So let me give you a good example. If I worked on plants that were drought resistant, so I've shared with you guys my idea about drought resistant soybean in Nigeria. So I work with plants that are drought resistant, and I find out that there is a soybean plant that is able to be drought resistant. Okay, no. I find out that there is a cactus plant. So everybody knows that cacti, they live in the desert. That means they can survive drought conditions. If I look at this cactus plant and I say, what is the gene that allows this cactus plant to be drought resistant, that allows it to live in the desert? The next step is, if I determine those genes, how can I take those genes out, put those genes into my soybean that is not resistant, and cause my soybean to be resistant? And the way that I would do that would be using the techniques of biotechnology. Okay, so biotechnology, I look at a gene, and by looking at a gene, I'm able to manipulate that gene, take that gene out, put it in a new organism. By doing that, I'm creating GMOs, and GMO stands for genetically modified organisms. And so many different organisms have been genetically modified, and they are changed, and they become completely new organisms because they now have another organism's DNA incorporated into it. Okay? And so in the slide that I gave you, it talked about cloning. And when you look at, I think I asked you the difference between cellular cloning and molecular cloning and reproductive cloning. The only difference between them is when you're talking about cellular cloning, you're talking about being able to take one organism like bacteria does, and from that one organism, you have a lot of different organisms. So cellular cloning means you begin with a unicellular organism, and from that unicellular organism, you're able to produce many different organisms. An example of cellular cloning would be binary fission. That's what plants do, okay? Now, the example of molecular cloning is the GMO that I told you about. So a transgenic organism, or a genetically modified organism, an organism that has the gene of another organism inserted into it, that would be an example of molecular cloning, okay? The next one, which I told you to think about, was reproductive cloning. And reproductive cloning is where you clone an entire organism. So when you clone an entire organism, and you guys are probably too young, but Dolly the sheep was the first mammal to actually be cloned in 1996. Okay, so Dolly the sheep was cloned, and on Blackboard I have some work that I want you to do. I'm going to use a few minutes, and I'm going to have you look at some information about Dolly the sheep. I'm going to have you research Dolly the sheep, because reproductive cloning brings about a lot of ethical questions. Can we clone organisms? So we know we can clone plants, which is what reproductive vegetation is, propagation. If you take a if you take a branch of a rose plant and you cut it into pieces and you grow it, a completely new rose plant comes out. That's cloning in a way, okay? That's vegetative propagation. But can you do that with an organism? What scientists were able to do as far back as 1996 was clone an animal. So not just are we cloning plants, but we're actually not just are we cloning prokaryotic organisms, we're cloning eukaryotic organisms. And Dolly was the first one to be cloned. Dolly lived until just a few years ago when Dolly died, and Dolly was a good example of a genetically modified organism. So GMOs are topical. You guys have heard a little bit about GMOs. There are things on there about advantages and disadvantages of genetically modified organisms. And so what I want you to do is put yourself in groups of 2. One person is going to do research on advantages of genetically modified organism. The other person is going to do research on disadvantages of genetically modified organism. And we are going to use the next about 10 minutes to do that research, and then you're going to try to convince each other genetically modified organism has advantages, so we should adopt it and use it, or genetically modified organisms do not have advantages and so we should not use it. Okay, and that's where we're going to end today's class. By the time you guys are done with that research, I will be there, because I want you guys to tell me what you think. Try to convince each other. Do your research. What are advantages of genetically modified organisms? What are disadvantages? Talk about that, and I will see you guys in a little bit. Okay?